Got that started. And okay. So let me see here. All right. Well, everyone, thank you for all joining us today for the HMSC Research Seminar. My name is Yi-Chung Chung. I'm the Academic Programs Manager at Oregon State University's Hatfield Marine Science Center here in Newport, Oregon. And I, as well as Michael Banks, will be your host of today's talk. Just wanted to go over a couple of logistics. So we have your mics, cameras, and screen share off, and we ask that you keep them off for this event. Please ask any questions you might have using the chat box found at the bottom of your screen at any time. We will answer any questions at the end of this talk. We'll be recording this event and it will be posted in a few days on the HMSC website's past seminar page. And I'll put that link in the chat. And before we get started, I do want to put out one announcement, which is next week we will not be having a research seminar in lieu of a special event that's happening next week, and that's the 2020 Luck Alliance Virtual Symposium on Sea Otters and Oregon's kelp ecosystem. And that's in partnership with OSU's Hatfield Marine Science Center and the Oregon chapter of the Wildlife Society. That's October 6th, 7, and 8 next week. And I'll put information in the flyer in the chat box so everyone can look at that and register. The keynote address will be Jim Estes on October 6th at 7 p.m. Okay, and with that, I will hand it off to Michael.
Hello, everybody, and welcome to the seminar, especially those in the class I'm instructing. And thank you, Alex, for joining us. So our speaker today is Dr. Alex Davis, who is a Liber Aero postdoc fellow in the Department of Biological Sciences, University of Alberta in Panda. So Alex is a marine spatial ecologist and an advocate for equity and diversity in conservation science, where she focuses on developing an interactive conservation tool to inform interventions of marine invasive species. So she works with multiple partners and management agencies to develop spatial priorities and quantify economic costs in managing the Indo-Pacific lionfish in the US Caribbean and tropical Western Atlantic territories, as well as the European green crab in the Salus Sea. She also studies equity and the lack thereof in conservation science, and specifically trying to understand barriers to assessing information and detangling colonial influences on how we practice conservation science. The neat thing about Alex was that before she went to University of Alaska Panda, she was actually a beaver, where she studied in Dr. Mark Hickson's lab for her PhD. And I noticed Mark is here today. I don't know if you knew that, Alex, but he's looking over your shoulder again. She used a combination of diving, GIS, and spatial ecology to analyze the distribution of lionfish in the Bahamas in her PhD. And before that, received a BS in marine science from California State University in Monterey Bay, where she worked in the seafloor mapping lab analyzing coarse grain sediment patterns on the California continental shelf. She also has a BA in human communications. So we are for a treat here, especially someone that was originally trained in communication. Thank you very much, Alex, and it's all yours now.
All right, can you guys hear me okay? Okay, well, thank you for that introduction. Thank you for having me. It's great to be back in Oregon, even virtually, doing a talk for OSU. So I'm just going to go through some of the stuff that I've been doing for the past couple of years, partly through my PhD and through my postdoc appointment here at University of Alberta. So first, I just want to give a big shout out. I As you said in the introduction, I am doing a whole lot of collaborative work with a whole lot of different people, and I had a lot of support going through my PhD and what I'm doing now. And so I just want to highlight some of those folks and some of the funding agencies that have contributed to this work and where I've gotten to today. So just a little bit of background of how I made it to where I am right now, sitting in Edmonton, Alberta, studying marine science. As Michael said, I started out as an undergrad in California in Monterey Bay, and I worked as an undergraduate researcher in the seafloor mapping lab. And so part of what I learned there was thinking about large spatial scales and how MPA networks can be set up and what goes into that process. Specifically, I got to help with creating seafloor maps based on different habitat types, and I did a lot of research on mapping the distribution of coarse grained sediment features. And so a lot of my undergrad work after I did my communications degree was spent looking at pictures of sand along the entire coast of California. As I went through my program, I was lucky enough to be at one of the schools, and I just did a talk there last week, or did a panel, and found out that the science diving program at CSUMB is one of the biggest in the nation now, which is really, really cool, because I was one of the first students to go through that. And part of the program, I got to learn how to be a science diver. You can see here in this picture, I'm wearing the full cold water gear, which is maybe not quite as bad as what you have to wear in Oregon right now. But after doing years of diving with 36 pounds of weight, I decided I didn't want to do that anymore. So I sought up some opportunities to do some warm water diving. And I was lucky enough to do a dive expedition to the Aquarius underwater reef habitat off the coast of Florida, and basically just fell in love with how easy it is to dive out there and the ecosystem. So my next step was to find an internship where I could do this more. And this is where I started to become connected to OSU. I was lucky enough to go and be a field assistant for Dr. Mark Hickson and his graduate students for a summer, and learned so much stuff and really, really liked the idea of doing diving for my entire PhD. And so applied to Mark's lab, got in, and was like, hey, sweet, I get to dive for, the next 5 years. This is really great. But, as an undergraduate, I spent a lot of time just thinking about fish in general, thinking about habitat and things like that. And part of being in Mark's lab was knowing about invasive species. We were fortunate enough to be some of the researchers on the forefronts of looking at invasive lionfish. And so for me, that was a new experience. And so as most people do nowadays, one of the things that you think about is, well, what is an invasive species? So when you go and Google invasive species, a lot of terms come up. There's things like invasive, you can now call them nuisance species, non-native, alien. You can even see things like range expansion or anthropogenically induced species. And so there's a whole lot of terminology going around it, all kind of centered around the same idea. You also get these pictures where they're pretty in your face, pretty wild looking, where you have words like harm and wanted. You even get these wanted posters. You get things like these kind of old B-movie type flyers that really try to emphasize the negative things that can come out of these invasive or introduced species. And you'll notice I have a few for Texas here. I just wanted to give a nod to my nieces and nephews who I hope are watching from Houston, Texas. So if you guys are there, look out for feral pigs. But even though there's a lot of terminology and a lot of stuff kind of coming at you for invasive species, I think some of the key points to think about when you're trying to pinpoint what invasive species really represent is these words here: species that can cause economic or environmental harm, specifically looking at biodiversity or the decline or elimination of native species. And so species will be introduced and species will have these range expansions. But what makes us want to study them are these potential outcomes of their interactions with the ecosystems and the species that we love. Now, invasive species are not a generalized or a localized problem. It's a global problem. And this map, I think, really illustrates, especially for aquatic and marine invasive species, how far reaching they can be. So you can see these different arrows and these different circles represent the pathways of invasive species moving from one continent and one country to another. And the size of these circles represents these areas where you have these high densities of marine invasive species. And so even if you're not in a location where there's a marine invasive species or an invasive species right in your backyard, there's potential you can go really, really close and have this be something that you can deal with. So after thinking about invasive species just as an individual component, I think the next step to do is think about how do you manage invasive species. Anytime there's some sort of threat or there's potential for compromise for something that we love, again, some sort of ecological system or some sort of species, we want to understand how to manage and mitigate the damage happening. And so I came up with 3 components for managing invasive species. And so the first is to just identify priority areas where you'll either find invasive species or where they're having these negative effects on the ecosystem or the native communities. Secondly, you can look at targeting densities for suppression. So it's not enough to just understand an invasive species. If you want to preserve a habitat or look after a species, you're going to want to remove the invader if it's having negative effects. And so knowing how many of the species you need to remove and using those for targets for control is really important. And thirdly, the actual act of removing invasive species is very important. So you need to understand effective and efficient removal practices. And a lot of this has to do with identifying your resources and understanding your system. How much time, how much money do you have, who are your personnel, and what gear do you have to actually remove these species from the environment. So I'm going to, for the rest of the talk, kind of keep referring to these 3 components for management. And the first thing I'm going to do is use a case study of invasive lionfish to kind of talk through how these components fit together for a management plan. So some of you may have seen this. This is the Indo-Pacific lionfish. It's invasive in the Atlantic and the Caribbean. So as in its name, the Indo-Pacific lionfish is native to the Indo-Pacific. You can see this distribution map here of its native range. They were first introduced off the coast of Florida in the late 80s, and since then have spread up the east coast of the United States into Bermuda, the Bahamas, the Gulf of Mexico, through the Caribbean, down into South and Central America. And so the geographic range of this species is quite broad. Now, going back to our components for management, thinking about priority areas. So lionfish have the potential to spread to quite a large geographic range. So what we want to do is quantify how they're affecting native communities, and then think a little bit more about specifically where these invaders are found. So a lot of work that went into studying lionfish at the beginning of the invasion was on these potential negative effects lionfish would have on native communities. And so a lot of work that my lab and other researchers did went into that and found that lionfish have high feeding and high growth rates. And this is both compared to native species in the invaded range and compared to their conspecifics in their native range. They're a generalist predator, which means they will eat pretty much anything that they can get their mouths around. And they've been shown to reduce native recruitment of fish up to 90%. They even have been shown to cause local extinctions of native fish, and they have really high fecundity, which means that they're going to be reproducing often and in large amounts. So strong negative effects on the invaded system. So my question when I went into my PhD was really concerned about where you find lionfish and what types of habitat they're associated with. And this is kind of going back to me looking at a lot of habitat as an undergrad. I was still interested in that, and I wanted to use lionfish as a study species to see if I could create habitat maps. So one of the questions I had as a graduate student was, can I use diver and satellite collected data to see what habitat features will help us predict where lionfish will be on reefs? So in order to do that, I did a lot of diving. Over the course of 3 or 4 years, I dove on 36 reefs and a bunch of different random points in the Bahamas off of the island of Eleuthera to get habitat data. And what that looked like was me going down diving and mapping by hand and counting the numbers of cracks, ledges, caves, and things like hollow coral heads, because that's where you often find lionfish and structure. They like to be tucked up in things and hidden away from things like currents. I also looked at the presence and absence and the density of lionfish. And so this is just giving me, is a lionfish there or is it not? If it is there, how high are the densities? And then I won't talk about this in this talk, but I also looked at the density of Nassau grouper, as the presence of large grouper has been thought to be a potential biocontrol for lionfish. So if you see a lot of grouper, maybe there won't be as many lionfish. The second component of this was looking at broad scale satellite remotely collected habitat data. And so in addition to these kind of micro scale metrics that I collected by hand while diving, I was also able to use the skills that I had from the seafloor mapping lab in order to create these habitat maps of the area. And so some of the things that that are commonly used to predict fish distribution are things like rugosity, which gives you the roughness of an area. You look at the straight line distance compared to all the cracks and crevices, and it gives you a rugosity measure. So for this one, you can see the green areas are really rugose, the purple are smoother, more likely to be sand. You also look at the highs and the lows, or the valleys and the peaks. And so these areas here in red, these are what are classified as peaks. Those are more likely to be things like reefs. Areas that are gray are flatter. Curvature is another metric used, and this is basically the slope of a slope. So how fast is the slope of this area changing? Those really sharp ledges are potential areas that are really sought out for fish refuge. Depth is another metric, and that is just depth in meters. And then the last one that I used was a classified habitat substrate map. And this breaks things into, you've got unconsolidated sediment, hard bottom, coral, and then hard bottom with algae. So using all these metrics, these 5 different broad scale habitat metrics, along with the micro scale habitat metrics that I classified by myself, I was able to use marine geospatial ecology tools and ArcMap combined with our software in order to create a generalized additive model. And what this does is it uses, my output is looking at lionfish distribution, and it uses all these different metrics into create my model. And again, previously I've used Nassau density, but I won't be talking about the results from that. So what this gives you is a visualization of where lionfish densities or lionfish are predicted to be. And so I'll show you 2 different maps based on some of the different metrics that I put into the models. So if you look at these maps, this one on the left is mostly a blue and red map. And so anywhere where you see red on the map is an area where high densities or high presence absence is predicted. Anywhere where you see blue, it's low prediction. And that will hold true for this map on the right as well. These blue dots represent an actual absence of lionfish, and the red stars are an actual presence. And so what you can see, this map here is based specifically on the broad scale seafloor metrics. And this one here is a combination of the broad scale remotely sensed ones and the fine scale hand collected metrics. But the main point of this is you can use habitat metrics to predict the distribution of lionfish, which is really great because if you're in an area where there's lots and lots of reefs and you don't have time to actually go out and look and see if lionfish are there, you can make an educated guess as to where you will find higher densities of lionfish. So thinking about what you'd want to do from a managerial perspective, you want to identify high priority areas. And 2 of the ways that we could do this are using these habitat maps that predict where we'll find high densities of lionfish, and then thinking about things that we want to conserve. And so specifically in reef systems, things like juvenile fish habitat and marine protected areas might overlap with those areas where we have high predicted density of lionfish. And then you can also think about things like ecologically relevant fish that are likely to be eaten by lionfish, such as parrotfishes, and economically important fish like juvenile grouper that you also want to protect. And you need to know the distribution of those in order to identify your very specific high priority areas. So thinking about the next step in our process for creating a management plan. The next one is to think about how to target densities for suppression. Do we want to remove all of them? Do we need to remove some of them? And how do you go about figuring that out? So just some background. This study did a survey of aquatic invasive species managers and practitioners, and 80% of the managers and practitioners said most invasions or the invasion that they're dealing with is occurring at a scale beyond resources for eradication. So eradication is not an option for most of them. However, 95% also said that they lack targets for suppression or for ecological recovery. So there's this mismatch between what managers know they can do and the information they have that they need to get there. So when is eradication the best approach? Well, looking at this schematic, let me just orient you right here. It kind of gives us a way to think about when we can think about eradication versus when that's not an option. So on this axis here, we have a geographic extent. So down at the bottom, very small geographic extent, and it increases as you go up. On the x-axis here, we have the different stages of invasion. And so every circle represents one of the invasive or non-native populations. And these squares represent the available resources you have for controlling the population. So when you first have an introduction, you have maybe a few individuals at a small scale, and you've got a lot of resources, so prevention is still possible. As the geographic extent increases and as the population establishes and grows, then you need to think about eradication. Okay, we can no longer prevent them from getting here, but we can keep up with the number that are here and effectively remove them completely from the system. And you keep moving along this axis to where the expansion of the non-native species and the geographic extent starts to outrun the resources that you have to be able to contain them. And then you get to this point where you completely basically swamped, you don't have enough resources, and the geographic extent and the abundance of the invader are way beyond what you can actually remove from the system. And another thing to think about too, once you get into these areas with range expansion and abundance increase, you no longer, it's often that you'll have multiple different agencies, governing bodies that are affected by the specific invasion. And so you get these patchworks of resources where you no longer have one that can contain it all. You have resources coming from different areas, but they still no longer encompass either the geographic extent of the invasion or the numbers that are there. So typically when you think about eradication, you're always targeting a number. We need to remove X amount from the system, but this paper by Green and Grosholtz, which is going to be published pretty soon, so hold your breath for that, talking about how to set ecologically based targets. And so thinking about, instead of how many can we remove from the system just to get a number, how many can we remove from the system to keep an ecologically sustainable system, or keep the system in a way that we want it to stay. And so this type of suppression is called functional eradication. And what one of its core principles is just thinking, how do we keep our ecological system functional, and what density do we need to remove the invader to keep it there? And so going back to lionfish, we know that they're found in multiple habitats. We are able to predictively map where their distribution is. We know that they have the capabilities to expand past where they are right now. This is a map of future spread based on sea surface temperature. Currently, the range is here, but they have the possibility to spread all the way down the coast of South America. And we know they have strong negative effects. So eradication is definitely not an option for lionfish on a broad scale anymore. So what's the next step? How do we figure out how many lionfish we need to remove from a reef in order to keep these reefs functional? So a study by Green et al. In 2014 did this exactly. Using surveys of fish biomass, predation rates of lionfish, and then looking at lionfish densities and body size, they were able to come up with a density threshold, which these reefs could still remain what they termed functional. So just kind of an example of what this looks like. So this is a results slide that shows the proportion of fish remaining after the start of lionfish removals for prey-sized fishes under 15 centimeters. And so what you'll see when I remove the, for the slide are, it's a time series with these 4 colors as the lines. And so this green and the yellow are treatments where lionfish densities were removed below this threshold. The orange and the red are treatments and reefs where lionfish were not removed below this threshold. So what you're going to look at is whether or not the final date shows that the proportion of fish remaining is above or below a line at the center. So as you can see, this is a study that was done from 2009 to 2011. And you have the green line and the yellow line, which represent the reefs where lionfish were removed below that threshold, had an increase in the proportion of fish on the reef. And so once they found those density thresholds and kept removing lionfish there, those reefs were still termed functional. And the interesting thing with this is that you don't actually have to remove all the lionfish on the reef. There's a gradient of densities that you can leave on. So it's no longer go out, remove every single fish. If you leave 1 or 2, you can often still have a ecologically functioning reef. So thinking about the next part, once we know our densities for suppression and once we know the areas that we want to focus our attentions on, the last part of this is understanding how to be efficient at removing the invasive species. And the things that I choose to focus on are things like time, money, personnel, and gear. So if you've ever done any sort of lionfish removal, you know it can be labor intensive and time intensive. Currently, the best way to manage the invasion and to remove lionfish is by hand, either using pole spears or hand nets. And so you have to go and remove every single fish individually. There is some work creating traps, which is a neat thing because you can get to depths beyond where recreational divers can reach. And so a lot of effort it goes into putting on lionfish derbies where they get a bunch of people to go out specifically in areas where maybe they're not allowed to fish regularly and do these lionfish culls. However, this is a lot of work. And this is just an example of like a regional effort to go out and do large lionfish culls. There's lots of people involved, lots of time goes into planning these and to managing these. And so anything that can be done to make these more efficient is definitely something that managers need to consider. So the project that I am working on is a regional study of what actually makes lionfish removal more efficient. And this study takes place in 3 areas in the invaded range. So the Flower Garden Banks National Marine Sanctuary in the Gulf of Mexico, 2 different areas in the Florida Keys, the Florida Keys National Marine Sanctuary and Biscayne National Park, and the Bird Island Reef National Monument off of St. Croix in the US Virgin Islands. And again, the data collection for this was a huge effort with lots and lots of people putting lots and lots of time into this, but it gave us a really cool, really rich data set to work with. So in order to get the data for this, folks went out and did bi-monthly dives tracking lionfish removal effort and success. And so you can see here, you go out and you do these very intensive lionfish searches to make sure you get everything on the reef. They have these lionfish catchers where you can store lionfish while you're looking for more. And you go out in pairs so that one person is taking notes and collecting the data while the other person is doing, or other few people are doing the actual removals. And so I'm showing this data sheet just because it has, it just shows the amount of information that went into every single lionfish removal. So for every single fish, you identify where it is in your transect, the size, what type of habitat it was located on, whether it was exposed or sheltered on the habitat, what behavior it was exhibiting, what time it was sighted, how many attempts you made at capturing the lionfish, what time you stopped trying to capture it, whether or not you got it, and what gear you used. There's also information here about the environmental conditions, such as time, visibility, current. So for every single lionfish caught, all of this information was collected. So this, like I said, was a huge effort. They're still collecting data now. I was able to go out on one of the surveys, which was really great. It was, but the data that I'll be presenting today is over the course of 3 years. So there were about 30 sites, over 50 volunteers, over 600 surveys. I think this number is way low now. I think it's in the thousands, but thousands of minutes underwater spent collecting this. And I just want to point out that there's a familiar face in here. So this is a trip I got to take last summer to the Flower Garden Banks to help do data collection. And Kevin, the DSO, was also on the trip. And so that was a really fun time to get back with some OSU divers after being in Canada for a bit. Okay, so after collecting all the data, the 3 response variables that we wanted to look at that we thought would help us decide how efficient lionfish removals were how likely it was the lionfish was to be captured, the proportion of fish captured on the reef, and then the time spent in capturing each fish. So in order to figure that out, we divided our variables into 4 aspects of removing lionfish. And so the first one is just things related to the invasive species, in this case, the lionfish. So how big was the fish? How high was the density where it was caught? What behavior was it exhibiting? And where was it on the reef? We then looked at the habitat, looking at depth, the vertical relief on the reef, and so that's basically the distance from the lowest point to the highest point. And then we also have information on habitat composition, and specifically we used coral cover and gorgonian cover. And gorgonians are these soft coral, fan-like corals that can be visually impeding and can actually physically get in the way of you removing species. We also looked at the aspects of the remover, so what type of gear was used. Most of the gear was spears, and so we restricted it to that in the end, the number of attempts made, and then we also looked at the experience of the remover, because every individual wrote down who caught what fish. So we were able to go and look at the number of dives they'd done before they started volunteering and whether or not they'd ever removed the lionfish before they started this project. And the last thing that we looked at was environment. Things like cloud cover, the current, and the time of day. And so just kind of an example of how this might look, thinking about how these things might affect lionfish removal. So habitat complexity. As a habitat gets more complex, there's just more things to get in your way. So the more complex the habitat, the more time or the less successful you might be at removing lionfish. Lionfish density, conversely, might make it easier to remove lionfish. If you spend time and you find 1, you happen to find 7, you spend less time searching, and so higher densities might make it more efficient to remove lionfish. Lionfish behavior can have an effect if they're out swimming, if they're actively hunting, as opposed to just sitting on the reef and don't notice you swim up on them. That may have an effect. And then the experience level of the remover. So here are some results, and let me orient you to this graph a little bit because I'm going to talk about 3 different panels of it. So this is going to be what is affecting time for removal. And so on the axis here, we have all of our different categories. So everything in red is related to lionfish. So things like density, size, whether they were sheltered, and what type of activity they were doing on the reef. Everything in this yellow-orange is related to habitat, so what the vertical relief was, the coral cover, gorgonian cover, depth, and then a combination of vertical relief and gorgonians. Everything in green is related to the remover, so how many attempts they made on the lionfish and what type of experience they had. And then the last one, the only environmental variable we ended up using was time of day. And so time for removal is a little bit different because everything that increases time for removal is something that we don't want. But if you look at this plot, there's a line down the center. Everything on the right-hand side is going to positively influence the variable. So specifically for time for removal, midday removals added time to what? Added time to removals. The other two will be slightly opposite, and I'll go over that. The only other variable that was significant in the model was coral cover, and that slightly reduced the amount of time it took to remove lionfish on the reef. So the other two, these are going to be opposite. So everything, but again, everything on the right-hand side of this line is still positively affecting, but now that's a good thing. We want it to be more likely for us to remove lionfish, and we want there to be a higher proportion removed. And so the things that were significant in these models were lionfish density, made it more likely and gave you a higher proportion removed. Lionfish size made it more likely for you to remove lionfish. Coral cover actually negatively affected lionfish removals, and so perhaps just having a more varied habitat made it harder to remove them. And then the last thing was depth was slightly significant and made it easier for you to remove lionfish. And so I'll go over these in a little bit different way just to clarify what all this means. So these are heat plots that show the probability of capturing a lionfish and then removing a high proportion. And so the two that really stood out for me were lionfish size and lionfish density. And for anybody who's captured lionfish, this is not necessarily this is not necessarily a novel idea, but it is nice to have it down in paper and have it quantified. So the larger the fish and the higher the densities, the higher probability you have of capturing the fish. And then for proportion removed, the thing that really made a difference was higher densities. If you have higher densities, you're more likely to remove a higher proportion. And so what does this mean for management? Well, lionfish size and density were important. The depth and then the coral cover of the reef were important, and time of day was really important. The interesting thing is that none of the variables related to the remover made a difference. And so I think this is really neat because then you can have folks who have less experience go out with those who have a lot of experience, and theoretically they will be just as efficient at removing lionfish. So when you think about planning a removal, you want to think about targeting lionfish that are big and in high densities. If you can, go to reefs that are deeper and have lower coral cover. And specifically, you want to target times that are at those crepuscular hours, so dawn and dusk. And what that looks like is, specifically, removing lionfish at dawn and dusk takes 1 minute less per fish to remove. And now if you only have one fish, that doesn't matter. But if you have reefs that have 20 to 25 fish on them, that significantly increases your dive time. And for those of you who dive, more dive time means more money, means more surface time. And so if you can target lionfish on those early and those crepuscular hours at dawn and dusk, you tend to be a lot more efficient. So next steps for this project. Well, because density was a really big factor in how efficient you are for lionfish removal, the next step is to figure out where lionfish are likely to be in high densities and where they're likely to be returning after you remove them. And so this is what I did for my dissertation thesis. And so the next step is to take it to the Florida Keys, where we have all this removal data. Sorry, Courtney, I made your image really ugly. After that, my next step is to think about how can this be expanded to other regions in the invaded range. And so we did the survey for Florida, the US Virgin Islands, and the flower garden banks, but we also have comparable data in areas like the Bahamas and the Cayman Islands. And because there's such potential for lionfish to expand to a large range, we want to know if this model is robust and can it be applied to all these different systems. And then lastly, thinking about what managers actually want. How can this be incorporated to current conservation plans where they have areas that they're already monitoring or spending a lot of effort and money into for things like protected areas and juvenile fish habitat? How can you integrate this lionfish removal into your conservation plan? Now, the overarching goal of this whole project is to see if this can be something that's used universally for multiple invaded systems. And so my next questions are, can this be used to model other systems? What components are generalizable? And then what components just have to be completely specialized for each system? Now, I'm lucky enough to be able to have secured funding to do just that. So I am a Liber Era Fellow with, I guess, affectionately called the Canadian Smith Fellowship. And so it's a conservation science fellowship. And I am going to take this kind of case study that we've done with lionfish and expand it to look at European green crab invasion on the west coast of Canada. So for those of you who don't know much about green crab, it's kind of a similar story, I think, to lionfish, where you have a highly fecund species that is a generalist predator and will eat a lot of economically and ecologically important species and outcompete them for food. In addition, green crab have the potential to be very devastating to different habitats, specifically eelgrass, which is a very important nursery habitat for a lot of fishery species, specifically salmon, which if you're on the west coast and the PNW in Canada, is very, very important, important species. Similar to the lionfish, green crab have the potential to have a really, really wide geographic range. And so you can see here in the blue, this is where they're native. Along here in the green is where they currently are invaded, and the red is everywhere they have the potential to spread. Where I'm interested in looking is in the Salish Sea. And so the Salish Sea borders the US and the Canadian border. And so for me, I think it's a really cool opportunity to kind of have this co-development of a management plan with multiple nations. Interestingly enough, there has been some work done on functional suppression of green crab. So looking at what densities do we need to reduce these to in order to keep our ecosystem functioning properly. And so reducing green crab does significantly reduce their ecological and economic impact. So despite the fact that in some areas where green crab are invasive, it's kind of a all doom and gloom scenario, on the west coast of Canada, specifically, we still have the potential to kind of mitigate the invasion, which is really great. So thinking back to the three components for management and switching it to the system for green crab, there's a little bit more of a patchwork of work that's been done here. There has been some identification of priority areas where they're monitoring for green crab arrival and areas where green crab have already arrived that they're doing active removals on. There's still a lot of work to be done on how the invader is affecting native communities and where specifically it will be found. Green crab are generally distributed by spawning, and so restricting ocean currents is not anything that's going to happen. So a lot of the removal and a lot of the mitigation has to happen at the subadult and adult stage. Like I said, densities for suppression have already been started. People have started studying those, which is really great. So I'm coming in at this area here where you're trying to understand efficient removal practices, identifying resources, figuring out who your personnel is, and figuring out best practices for removal. Some of the reasons I'm really excited about this is just because it's a really cool collaborative multi-nation, multi-state effort. Like I said, the Salish Sea borders both Canada and the US, but green crab are not limited to that area. Along the West Coast specifically, they're in California, Oregon, Washington, all the way to Alaska. And so there's a lot of collaborative work going on understanding how green crab are moving through this system. It's also multi-agency, and so each of these agencies are going to have different priorities, different resources, and different availability of how they can manage this invasion. And it's also at multiple stages along the West Coast. So some areas they're established, some areas they're completely green crab free, some pop up and they're able to eradicate them right away. And so I think this is a really cool stage of the invasion and the invasion process for me to get into because there's so much going on. It's really neat. And kind of the last thing I want to talk about is just thinking about how to make this accessible to managers. There's a lot of information that can go into this. And so using things like prioritization tools that are typically used for designating conservation areas is one way that we've been thinking about integrating all of this information into a user-friendly interface for managers. And so things like Marksan, Zonation, and Prioritizer are these web available interfaces that managers can go in and kind of say, I have X amount of dollars, I want to preserve this amount of space, and I have this amount of time, and it can give you an output and help you create a management plan. And introducing invasive species into those conservation management plans is not something that has been done yet, but I think it goes hand in hand with what most conservation managers are trying to achieve. So that is all I have for now. And if anybody has any questions, I will be happy to take them. I'll stop sharing. Oop.
Alex, there are a number of questions in the
Okay. I saw those popping up. Okay. Should I just go through and read them, or?
Sure.
Okay. Let's see.
It starts off with the one at 405. Does lionfish removal involve any risk for the divers regarding spines and venom? And if so, does that factor into weighing the cost of removal?
Mm-hmm. That's a good question. That I think is one of the probably most important parts of training for lionfish. I made it through my entire dissertation without ever getting poked by a lionfish, and I feel really lucky. Some people get poked and some people have bad reactions, some don't. And so the training that goes into proper care and handling of lionfish is definitely something that needs to be factored in. I wouldn't say it's a huge of risk as it sounds like it is, knowing that you're handling a fish with venomous spines. So other people can probably negate what I said, but I never got poked, so I'm a little bit more less worried about it.
Oh, great. Next question is from Charles Nye. Is there any effective bycatch during these events by removers and fishers?
No, that's a nice thing, because there may have been instances where you actually accidentally spear another fish, but because you're actually going down and individually identifying the fish and catching that one on its own, bycatch is not a problem.
It looks like Philip Cart has said minimal risk if containment device is used.
Yeah, they have these great PVC pipes with it's like a one-way tube, and you stick the pole in there with the lionfish, and when you pull it out, it's got these rubber flaps that pull the lionfish out, leave it in the tube, and your spear is clean. So really cool innovation that somebody came up with that I think is a good safety protocol.
That's after it's been speared.
After it's been speared, yes.
Then Kevin has posted something about deeper lionfish drop traps are being found to be lionfish specific. Mm-hmm. You find that true?
I've only read the paper about traps once. I know there's a little bit of bycatch in those, because as with any traps, there's going to be some sort of bycatch, but they're still in the preliminary phases, and I think the results are pretty good.
Kurt Ingman have green crab populations shown the same pattern of exponential growth along the invasion frontier, with lower or negative population growth rates in the interior, where invasion is older?
Oh, Kurt. Let's see. I'm trying to find what I could do. I would say no, but I could be wrong. I'm trying to think of the population along the West Coast is specifically in Canada is so patchy. And so there's not the consistent growth that you see kind of like that you expect with an exponential growth. That may be different, because I know like on the East Coast of Canada, it's I want to say riddled with green crab. And so there it's more like the exponential growth, but on the West Coast, I don't think so.
Okay. Laura Jacobs has asked, do you involve any indigenous nations in your research, especially for your new work with the green crab?
So that is the goal. So I am working specifically with a mentor at Departments of Fisheries and Oceans. And one of the things that he participates in, the Now I Am, is kind of this like green crab invasive species network along the west coast of Canada. And that does include indigenous nations. And so that's one of my goals, is because there, especially like on the island of Haida Gwaii, where the green crab removal effort is all through the indigenous nations. It's really important to have that every aspect in. And this is one of the, like I said, this is one of the reasons why I'm really excited, because there's so many different managerial bodies with all who have different goals. So including First Nations is kind of key to me understanding the different levels of removal effort that need to go into the system.
Looks like Phil posted something about Steve Giddings' NOAA non-containment trap, and thank you for that, Phil.
Yeah.
And then, Ben Wishnick has asked a question: when do you anticipate having the European green crab work done? Interested in potentially applying this in Alaska to prioritize areas to survey.
You know, I anticipated having a lot more done this summer, but unfortunately I was not able to get out and work with any of my partners on the coast. So I will keep you updated on how fast this is starting to move. At this point, I'm kind of in the networking and collecting data from partners who've already been doing some work stage. So hopefully it gets going. It picks up speed soon, but yeah, my start has been disappointingly slow, as I'm sure a lot of people's summers were.
Danielle Gillies, for either the lionfish or green crabs, is there any fisheries market support for catching and selling the invasive species as an effective way to support control? They read lionfish are tasty and thought they saw a cookbook photo in your presentation.
Yes, so I don't know that there's a whole lot of interest in green crab specifically, but they have discussed having a fishery for lionfish, although I think the latest chatter is that it's not necessarily sustainable, but it is edible and it is tasty. And some of the efforts for encouraging people to go out and catch lionfish were paying the same amount that they would for lobster in certain areas. And so one encourages folks to go get lionfish and it discourages poaching lobster. So there have been initiatives like that where you can actually get money for catching lionfish, you can eat lionfish, and yes, they do serve it in some restaurants with like an invasive species menu angle as well.
But not so much for the green crab.
I haven't heard much on that, and crab is not something I've ever been interested in eating, and so I'm not willing to go out into lionfish. I was willing to eat my study subject. I'm not so sure about green crab.
You should look into a soft shell green crab fishery, folks.
Yeah.
Michael's posted a question. Please provide more detail about group or potential control for lionfish, as well as any natural predator of green crab.
So there has been a lot of back and forth in the literature for grouper as potential control, but some of the things that have been discussed, so grouper are just a natural competitor for the same food that lionfish will eat. So theoretically, if you have more lionfish or more grouper on a reef, they will be in direct competition with lionfish. There has been some kind of back and forth about whether or not you can get grouper to eat lionfish. I think there's been an instance in the Caymans where if you catch a lionfish, you can feed it to a grouper. However, there is limited, I think with both grouper and things like sharks, limited success in actually getting them to adjust to having lionfish as a source of food. So the jury is still out on that. For green crab, as far as I know, have all the same type of predators that the native crabs on the shores would. And so I'm not sure if they are less palatable than some of the native crabs. That is something that I need to do a little bit more work and research into. But I think the main issue with green crab is not that they're not getting eaten, it's that they're out competing native species for resources and then destroying habitat. Oh, one of my lab mates says small green crabs are fried and used as croutons in New England. So if you get them when they're small, you can fry them into croutons. That's really cool.
Nice. Charles Nye, an environmental DNA meta barcoding work being done to accompany predictions for lionfish habitat occupation. Oh, any environmental DNA meta.
Yes, but I do not have the expertise to talk about how any of that is working. I know there have people who've done some of that, but I am sorely behind on any sort of genetics information, so I wouldn't want to speak on what that has found.
Okay. And Kurt Ingman says Liber Eero should give you an extra year. I think we all agree with that one. Another question from John Stepanek. Is there much public interest in green crab removal that people can eat? I think kind of somewhat addressed that already. You think it's possible to achieve functional eradication without needing regular active management to maintain a density threshold?
I don't know yet. That's what I'm hoping to find out. So green crab are, you catch them in traps. And so the hope would be is that you could set traps out relatively infrequently and keep your densities low. But I think it would also depend on what your starting population is. And I think specifically for green crab, the recolonization rate. And so if you have an area where you're just going to get a large influx of green crab, regardless of how many you remove, you're going to have to be actively removing them. Whereas if you have a place maybe that only gets small influxes or infrequent ones, then you can have less active removal. And so that's part of what I'm trying to, I think a lot of the work that's going into green crab removal is trying to figure out, how do we figure out where they're more likely to occur and where you're going to get these places where they'll recolonize over and over.
Let's see here. I'm looking at, looks like Vance M, Mitch Vance. The green crab are a bit too small for market potential. Yeah. Seafood Watch has done a boost on lionfish fisheries. Oh, it looks like Kevin has posted soapbox chance. Successful invasions require completion of life cycles by multiple founders. What if all post-invasion resources were directed to routine pre-invasion environmental monitoring? Sounds like more diving to me.
Yeah. It could be. I don't know specifically for green crab. One of the things we're really actually, speaking of like going back into monitoring, is going out and looking at all these reefs where we have done removals for lionfish and seeing what they look like after nobody's been diving on them for potentially months. So yeah, I don't know, Kevin.
No, it's valid.
Mm-hmm. Yeah. And so I think one of the things with that, like the pre-invasion environmental monitoring, is really understanding where you're going to find the invasive species. And so picking those places where the monitoring would actually make sense and it would be likely that you'll see the invasive species. And so if you're just going out and monitoring anywhere willy-nilly, that might not be successful. But if you can predict where you think or whatever your invasive species, if you can predict where you think it'll be likely to occur, then pre-invasion monitoring is definitely a good idea. I think that's one of the things with green crab. They're doing a lot of pre-invasion monitoring because they're not sure where they will end up. And the one that they spotted at Haida Gwaii was the first one that they saw. They saw one, they removed it, and now they're still green crab free. And so things like that. So there's potential for that. It's just understanding where to do the monitoring efforts.
Well, that's great. I have one final question, if maybe we can end on that. We're at 4:30 here, and so I was taking some mad notes. And I was question about the model and what you're using. Does the model take into consider, or are you guys looking at incorporating any life history of the lionfish itself, the reef fish in the habitat that the lionfish are occupying, as incentives for looking at densities? So if the lionfish population is targeting, and I have very limited knowledge about the life history of reef fish, but are there seasons where the lionfish will congregate and target say a bloom or a peak growth or peak density related to mating.
Hi, Libby. Sorry, my sister just popped up.
Or the
yeah, so reef fish will go through recruitment seasons, and so typically during the warmer summer months, and so that's when you'll see a lot of vulnerable small fish get eaten, and that's when you see you'll see maybe like you said, congregations, lionfish spending more time on those reefs where you have these large recruitment, large densities of recruit fish. And so in the model specifically, we don't look at life stage. Size is something that we do specifically for lionfish. And one of the next things that we're looking at is what's going to predict high densities and high recolonization rates. And prey fish will definitely be in that model. And so looking at reefs where we have high densities of prey fishes or high densities of smaller fish of those important species will be areas that we may get more likely to have higher densities of lionfish, or more likely lionfish to come back because there's an abundance of food or abundance of fish in the size range that they like to eat.
So annually, is there like a temporal cycle that?
Yes. Yeah. Yeah, for both lionfish and the prey fish species, you'll see bumps during the summer months.
Great, thank you. Well, that concludes our seminar today. Thank you all for joining us. Alex, thank you again for talking to and sharing what you've been working on in the past and what you're working on now. And I encourage everyone to check out our past seminars. This one will also be put on that web page. Alex, do you have any last remarks, or Michael, do you have any last remarks for the HMSC community?
Just thank you everybody for coming. I'm excited. This is the first time I've gotten to share my work because all my conferences got canceled. So that's really fun. And if anybody has any questions about applying to the Liber postdoctoral fellowship, please email me. So it's a really cool opportunity. And if you don't mind working in Canada, it's a great place to be. So
Alex, would you be able to put your email address in the chats for folks?
Yeah.
And I don't know if I can maybe pull up the Libro page, and I'll post that as well.
Oops, that's
Alex,
not raw. Hold on. There we go. I always spell Alberta wrong.
Alex, thank you so much. It was a very fun hour with you. Really inspiring.
Yeah, thank you.
See, I think this is the California Libro program. Let's see if I can find the. No, that's it, right? That is it.
Mm-hmm.
Okay.
Yeah, it should be liberaero.ca for Canada. Hi, Megan. Hi, John.